I'm going to introduce our recent work on reference panel guided super-resolution inference of high-C data. I'm Yanlin Zhang, a PhD candidate at McGill University. I conducted this research with Professor Matthew Blanchet. I'll first give a brief overview of 3D genome. As we are living in a three-dimensional world, the genome also has preferred 3D structures. At different scales, it has different structural preference. For example, there are AB compartments, topologically associating domain, and their quantum loops. Understanding these structural preferences is very important because they play essential roles in different types of biological processes. The genome is very small. We cannot use a microscope to visualize the genome in 3D space directly. The most widely used approach is Hi-C, which is a sequencing-based approach. The output of the Hi-C experiment is a contact map. It is indeed a matrix where X's correspond to genomic positions and the entry RG star value. This value indicates the contact probability or contact frequency between DNA fragment R and the DNA fragment G. A very important concept in Hi-C experiment is data coverage. It refers to the number of contact pairs captured in a Hi-C experiment. It can range from several hundred millions to billions of contact pairs. And we can have high coverage Hi-C matrix or low coverage Hi-C matrix. Here are two examples. The Hi-C contact map is pretty large. Let's suppose we are working on 5 KB resolution. For the whole genome, the matrix is as large as 600,000 times 600,000. Even for a single chromosome, there are more than 50,000 columns and rows. So for Hi-C contact map contains several hundred million contact pairs. It is indeed a very sparse matrix. If we compare the low coverage version to a high coverage version, we will realize a lot of signals are absent. As mentioned, there are different types of 3D structures. Each of the 3D structures correspond to a special pattern on the Hi-C contact map. So if we have a Hi-C contact map, we can detect such patterns to identify different types of topological structures. How to detect topological structure from Hi-C contact map is not easy. This is a distribution of contact pairs of published Hi-C experiment in the past decade. Most of the Hi-C experiment contains around 250 million validated pairs. Actually, they correspond to a Hi-C contact map like this. If we compare this Hi-C contact map to a Hi-C contact map of the same region, but sequenced at pretty high coverage, we will realize a lot of signals are absent from such low coverage Hi-C dataset. So that means the problems of Hi-C based study is we don't have enough high coverage data. In a Hi-C based 3D genome research project, we always want to have a Hi-C contact map that contains as small contact pairs as possible. However, the Hi-C experiment is hard and expensive. In most cases, we only have a low coverage Hi-C contact map. To fill the gap, we can try to predict an enhanced contact map based on the low coverage Hi-C contact map. This task is called contact map enhancement. Our primary contribution in solving this task is we propose a approach to make better use of existing Hi-C contact maps to improve contact map enhancement. In recent years, there are several tools already proposed to address the contact map enhancement task. Most of the tools are deep learning-based approach. As time passes by, we can observe newer models usually have more layers, and some models also introduce the generative adversary training to improve their prediction quality. We can summarize All of the models is a single content map-based content map enhancement approach because all of them use a single study sample as input to make the content map enhancement. Although there are several tools already proposed, most of the tools do have limitations. They perform well in enhancing a typical coverage high-C content map, but most of them cannot enhance a pretty low coverage high-C content map. As you can see, there are signals that absent from the low coverage input, and such missing signals are also absent from the enhanced content maps. Also, most tools use image super-resolution algorithms to do the content map enhancement. They do introduce artifacts. In a high-C content map enhancement task, we are aiming at addressing the data insufficiency issue. Although the 3D genome conformation is cell-type specific, when we pay attention to a small region, we will realize many local structures are conserved across cell types. Here is an example. At this particular preparation, we can observe the existence of topological associated domain boundaries at two different cell types. As mentioned, we don't have enough high coverage data, but we have many low coverage data. This indeed creates an opportunity for us to contribute to the content map enhancement task. We have introduced a reference-enabled high-C analyze framework last year to address the data insufficiency issue. The core idea is we can augment the input with additional existing data. Here is our generic framework. It contains an encoder and a decoder. The network takes the study sample and the additional reference samples as input. The encoder will project the input to later embeddings. The decoder takes the combination of the embeddings for the study sample and a transformation of the embeddings for the reference panels as input to make the final predictions. How can we perform the transformation? We use an aggregator to aggregate information from the reference panel such that we assign more weight to samples that are more similar to the study sample. Last year, we introduced the reference panel guided topological structure annotation of high-C data to annotate TAD and loops from an insufficient coverage high-C content map using additional reference samples as input. This approach outperforms existing approaches in TAD and loop annotations. This is an overview of our RegSys SR model. We used the UNet architecture with 3 levels as a backbone to handle dense, large path predictions. To take the reference panel into consideration, we adapted the encoding part of the UNet a lot. The encoding part now taking study sample and reference samples as input. Embeddings for the first 2 layers are used to make predictions. The third level is used to compute a weighted sum for the aggregation of the first 2 levels. This aggregation is a contextual attention module, except that we compute the attention output at 2 other layers. The decoding part will take the concatenation of the attention and the embedding for the study samples as input to make final predictions. RegSys SR is a deep learning application. We need to train a model before using it. We created a reference panel of 30 high-C samples. On the following previous publications, we used the GM12878 cell line as the study sample. We defined the target as the combined content map and the input as a series of downsamplings of the combined high-C content map. During training, we used L1 loss to train our model, and we split data according to chromosomes. We use chromosome 11 and 12 as validation set, chromosome 50, 16, and 17 as testing set. we use all the other chromosomes as training set. In addition, we pre-train the encoder with contrastive learning. The goal of contrastive learning is to encourage similar samples to have similar embeddings, and different samples to have an embedding as far as possible. And to achieve this goal, we use the cross entropy with negative samples in batches as the loss function. We compared RegSys SR against different tools, including Deep Hi-C, Hi-C Plus, Hi-CNN, in enhancing Hi-C data of multiple cell types and sequencing carriages. We used the structure similarity index, Hi-C rep, Pearson correlation coefficient, and L1 loss to evaluate predictions. In addition, we also studied how can we benefit from using enhanced contact map to perform downstream analysis, for example, the loop annotation and the tag annotation. We first enhanced a typical coverage Hi-C contact map using different tools. Here is an example of enhanced full coverage and low coverage Hi-C contact maps in a small region. As you can see, all the enhanced contact maps are very similar to the ground tools. They achieve very high structure similarity index. We then evaluate the enhanced contact maps by comparing them against the full coverage data with Hi-C rep. Across all chromosomes, RegSys outperform other tools slightly in their contact map enhancement. In addition, we computed the diagonal-wise Pearson correlation coefficient (MEE) and SSIM between the enhanced contact map and the full coverage contact map. All confirm that all enhanced contact maps are very similar to the full coverage Hi-C contact maps, with a slightly advantages observed on RegSys enhanced contact maps. All contact map enhanced tools are trained and evaluated on Hi-C data for the GM12878 cell line. Here we evaluate how the trained model performed on unseen data of other cell types. We applied each tool to enhance low coverage contact maps of two cell lines and compared their enhancement to high coverage contact maps. These Hi-C rep scores indicate that RegSys SR outperform other tools in enhancing Hi-C data of other cells. Hi-C experiment is usually conducted at different levels of sequencing coverage. Here we studied how the contact map enhancement tools perform in handling Hi-C data containing different numbers of contact pairs. We first created a series of downsampled GM12878 Hi-C data, and we enhanced these downsamplings with each tool. We evaluate the quality of the contact map enhancement by comparing the contact map enhancement to the full coverage data with Hi-C rep. The performance of all tools are equally well in enhancing data with more than 500 million contact pairs, but for low coverage data, we can observe that RegSys consistently outperform alternative tools, with larger advantages observed in very low coverage data. The goal of Hi-C contact map enhancement is to ease downstream analysis. Here, we evaluate how can we benefit from analyzing enhanced contact maps. We first compare the impact on loop annotation. We used MUSTAS to annotate loop from high coverage, low coverage, and enhanced contact maps. We also include our previous tool, RegSys, for loop annotation into comparison. RegSys is a tool that can detect loops directly using a reference panel. RegSys SR can enhance a contact map using a reference panel. So we compare the loop annotations to charped data and high-chip data for different proteins. RegSys SR identified the most charped or high-chip supported loops. CTCF is a very important element in maintaining loops, so we often use it to evaluate the accuracy of loop predictions. This figure shows that RegSys SR outperform alternative tools significantly. We then studied how the enhanced contact maps facilitate tag annotation. We use the robust TAG to detect TAG boundaries and compare boundary annotations against the CTCF binding sites. We observe boundary annotations from RegSys SR enhanced contact maps are more enriched for CTCF binding sites. To summary, we propose the RegSys SR, a reference panel-enabled tool to enhance high-C data for given study sample. In contrast, existing contact map enhancement algorithms are purely study sample-based. In this research, we demonstrate that RegSys SR works well across different cell types. It is robust to sequencing coverage, and it can also improve tag and loop annotations. Finally, I want to take this opportunity to thank all group members at McGill University and their funding agency, including Genome Canada, Genome Quebec, and the FRQNT, who support my research in the past several years.